Hello, everybody, and welcome to this module that's going to focus on the laboratory aspects of testing. So some of the technical issues going all the way through from diagnosis of CML to monitoring response to treatment, picking up those patients who have suboptimal treatment and may have resistance-associated mutations. And we're going to be looking at this in the context of what is the state of the art, essentially, and also what is the diversity of approaches that are used around the world and how technology may be adapted for different situations. We'll discuss where we are at the moment, future directions, and some of the very real practical problems that may be encountered. So I'm going to start off by looking at molecular monitoring, the international scale for B-cerebral measurement, which has really helped to bring everyone together around a sort of common approach to molecular monitoring, something which didn't exist before. Talk about the limits of current approaches and the standardization that's really been going on internationally for many years. I'll also mention digital PCR and the importance of detecting atypical BCR-ABL fusions and the consequences of what happens if these are missed. Sue Branford in Adelaide will be focusing very much on resistance. So a lot of interest here in detecting mutations, how those mutations may be detected, whether Sanger sequencing, next generation sequencing approaches are better, their applicability, and also the very important and exciting new emerging world of genomics and how this fits into routine testing for CML. So these are basically two talks that are focusing on the state of the art, perspectives from both Europe and Australia. We're then moving on to Dr. Neelam Varma, who's the head of hematology at the Postgrad Institute for Medical Education and Research in Chandigarh in India, where we really begin to understand some of the problems in applying these approaches worldwide. Resources are more constrained and patients tend to be picked up rather later in their course of disease. So she will describe the testing and treatment landscape in CML in India, and in particular the use of the Cepheid machine for molecular monitoring, which is one of many possible approaches, but may be particularly applicable to certain environments. Then finally, we'll have Jerry Radich from the Fred Hutch in Seattle, who'll be giving his unique perspective on new ways to improve molecular monitoring in resource-constrained environments, both low-tech and high-tech solutions. Low-tech going all the way from no electricity through to the other systems where you may collect samples locally and send them into a central lab abroad for analysis. Very important work, and a fascinating talk, which he's found particularly rewarding and we're all very much interested in. So I hope you find this module interesting. We have a range of approaches. I'm sure all the speakers are going to be very happy for any follow-up questions if there are any. Thank you very much.